Good morning, everybody. My name is Matthew Savallo, and I'm here to talk to you about SARS-associated coronavirus. So SARS, what is SARS? SARS stands for Severe Acute Respiratory Syndrome, and that name is derived from the effects that the symptoms have on the body, which we'll talk about in a couple slides. SARS is a virus. It's a coronavirus, kind of like the one responsible for the common cold, which makes up about, 30% of the common cold are coronaviruses. But this one's special. SARS are the only coronavirus to cause high mortality and morbidity in humans. And generally, they are spread airborne in the air, and there is no known cure. So the symptoms. What are the symptoms? Initially, there's flu-like symptoms. This kind of made it difficult to detect when it outbroke in 2003. They're initially fever, muscle fatigue, and chills. 20% of people who are infected do have diarrhea. Everyone likes to talk about that. And then in about 3 to 7 days, you'll obtain a dry cough, which helps to spread the disease pretty well. That'll lead to pneumonia, heart or liver failure, and then the big boys, pulmonary edema and diffuse alveolar damage. Pulmonary edema being a buildup of excess fluid in the lungs, and diffuse alveolar damage would be damage to the alveolar sacs, which makes it difficult to breathe. And here we have a healthy lung, and then a lung infected, or a lung that has gone through pulmonary edema with, obviously you can see excess fluid in the lungs. So outbreaks. So like I said, SARS started in 2003 in Hong Kong, China, and in a year it spread to 24 countries in North America, Asia, and Europe. Luckily, it was contained right away. It was spotted and detected, and people were quarantined, and the people who had the infection were quarantined. They were contained. They were isolated to not spread the disease any further. Today, that people in China sometimes wear the masks that can be associated with the SARS outbreak in 2003. The World Health Organization calculates that about 8,980 people were infected and 774 people died, making the mortality rate about 10%. In America, only 8 people were infected, and it was contained and stopped. So how is it spread? What is its contagiousness? Well, SARS is spread airborne in close person-to-person contact. So airborne through droplets of infected mucus or saliva that you can cough or sneeze out of the body. That's why that dry cough makes for such good contagiousness. Those droplets can then have the coronavirus, bind to mucous membranes on the eyes, nose, and mouth of a healthy person. And that being said, if I touch something that has the virus on it, the droplets on it, and I touch my nose, my mouth, my eyes, then that'll seep in and to my membranes and bind and make infecting me. But like we said earlier, there are no known drugs, cures, or antivirals for the virus, and antibodies are recommended for this because while your immune system is fighting off this virus, you need to be able to protect it against something else. So what is the molecular structure? Like we said, it is a virus, but it's an envelope virus. It takes the membranes from the cells that it replicates in. It is single-stranded RNA, has single-stranded RNA, and some proteins, the main ones being spike glycoproteins, which protrude from the envelope of the cell and are responsible for the selectivity of the virus. Envelope proteins, which are along the envelope of the virus, and nuclear capsid proteins, which we'll talk about in the next slide. In vivo replication. So in the cell is where the virus replicates. It binds to the receptors and enters into your cells. There, N, the nuclear capsid, and S, the spike proteins, activator protein, or AP1 for short. This protein is responsible for cell proliferation or growth, differentiation, and apoptosis or cell death. And it's also responsible for the interleukin 8 induction, which is responsible for phagocytosis and part of the immune response. That being said, it also induces MAPK and ACT, which are responsible for apoptosis, MAPK being a pro-apoptosis protein, and ACT being an anti-apoptosis protein that regulates the apoptosis in the cell. Because SARS infects the respiratory tract, there is an increase of the IL-8 cytokines, which are responsible for the inflammatory response, and that inflammatory response is going to increase. This slide here just demonstrates the in vivo replication where the receptors, this virus binds to the receptors on the edge of the cell and it enters the cell, uncoats, and sends out its RNA to be replicated. It then either replicates and goes on to infect more cells, or induces the AP1 and IL-8 pathways, as well as the controlling apoptosis. So where did it come from? What's the origin of SARS? So SARS is a coronavirus, but it is an original coronavirus to the human body, meaning that there is no coronavirus like SARS that has been infected in humans before. So we believe that it actually has come from another species, being a civet cat or a horseshoe bat, where that coronavirus in those animals was able to cross species and infect humans. But there is still no confirmation on which one animal virus caused the outbreak. And so why we talk about this in a bioterrorism class? Well, it can be used as a bioweapon. It is dangerous, and the CDC notes that, saying, on October 5th, 2012, the National Select Agency Registry Program published a final rule declaring SARS coronavirus a select agent. And what is a select agent? A select agent is a bacteria, a virus, or toxin, could be a bioweapon, with the potential to have a severe threat to public health and safety. It's pretty dangerous. We also do not have a cure for the virus, so your immune system just has to work. And there are studies done where you can actually engineer this. It could possibly be used as a bioweapon. We have not seen it been used yet, but the Department of Epidemiology at North Carolina University, or University of North Carolina, did a study where they were able to replicate and isolate recombinant SARS coronavirus. And it spreads pretty easily. It spreads like the common cold, just coughing onto a table and someone else touching it. It's pretty disgusting, but that can spread the disease. Anything with breathing it in, talking to somebody, is pretty easily spread, and it has a potentially fatal infection. So, like we said, 10% of the people infected in 2003 died. If that was a bigger population, if we were not able to contain it, 10% of the population is a lot of people. And it's uncontrollable, though. Uncontrollable meaning it wouldn't be a good bioweapon if you're trying to isolate a specific country, city, town. If it is spread, it's going to spread wide. That being said, anyone who is looking for doomsday or to bring about the apocalypse would kind of use this. Anyone looking to kill as many people as possible, are looking to eliminate a lot of people, could use this if it was not to be caught and contained and quarantined and all the stuff we did in 2003. And if it's not, then you can infect the whole world like in this game here. Thank you very much. Have a great day.